pleased to come first time here and visit the institute. And my lab is working in the area of skeletal muscle and heart muscle. And we do a lot of work both in vitro and in vivo. I'll try to kind of limit it to about 2 topics, or most, one related to heart, or a couple related to heart, and then a couple to skeletal muscle. So the system that we've developed that's mostly used for our in vitro culture of engineered heart and skeletal muscle is built on using this technology, which is generating a frame that's here shown in this opaque white, which is basically just a porous polymer that can anchor hydrogel and cells. And this anchorage at the ends or on the sides basically makes boundary conditions that then allow cells to align and remodel hydrogel. And for heart and skeletal muscle, that can introduce the orientation of cells, or you can introduce pores, as shown in these networks, that would provide more of a diffusional or overcome diffusional barrier and provide the ability to make thicker tissues. The cells we use for across many years include primary cells, and also as iPS or stem cell, pluripotent stem cell derived muscle and heart cells. So we use primary neonatal right ventricular myocytes, and I'll show you some work with that. We use mouse cells, pluripotent stem cells, when they were available and very popular. But as soon as the human cells have come into play, we've started working a lot with human pluripotent stem cell derived cardiomyocytes. And also for skeletal muscle, we use primary rat and human muscle cells, or myoblasts, as well as pluripotent stem cell derived muscle progenitor cells. Okay, and we published like a few papers on this, but I'll try to limit it to the most recent work. All right, so one of the systems that we use that I've shown here is basically what we call cardio bundles, which are these 2 cylindrical tissues, which I hope you can see. All right, so you can see my. So these 2, my mouse, so the 2 cylindrical tissues, basically, as I said, that are anchored within this porous frame, made basically starting from fibrin-based hydrogel with mixed cells of different kinds, cardiomyocytes in this case. This, after 2 weeks, spontaneously contract, as you can see. They are cultured on a rocker under dynamic and free-floating conditions. And this, we've shown, really helps with maturation of cardiomyocytes in these tissues. If you look at the staining of these tissues, you can see that in the longitudinal sections along these cylindrical axes, long axes, the cells are aligned, cross-striated. In cross sections here on the bottom, they are basically, you can see just, again, cells being aligned and filling the space around these cardiomyocytes shown in green, fibroblastic cells stained in vimentin. And this is, how these look, and they look very similar whether it's rat cells or human cells. Now, we've shown that specifically when these are cultured over a few weeks, they can actually achieve functional properties that are, almost comparable to those of adult tissues. And in particular, force that's generated per unit cross-sectional area, specific force reaches about 70 millinewtons per millimeter square of tissue. And the velocity, how quick action potentials propagate through this tissue, are about 50, 60 centimeters a sec. And so these tissues, which are the most mature when done with neonatal right ventricular myocytes, also show the progressive exit from cell cycle. So what you're seeing here, if you look at cross sections and label the cells with EDU for the cells that enter the S phase or start to replicate their DNA, you can see that with time, and shown on the right also, the number of cells that are in cell cycling decreases, and after a few weeks reaches really below 1% basically for cardiomyocytes. And so this exit in cell cycle is another sign of maturation. These cells also start expressing T-tubules as another maturation marker, and kind of match after 5 weeks under specific conditions, can match what you've seen in 5-week-old heart from rats. Right? Okay, so why are we interested in this and in these tissues? Well, one of the I'll give you a couple examples, but one of the things that we do is really have interest in looking in some of the rare diseases. In particular, diseases called rasopathies are the ones where there is a gain of function in MAP kinase pathway. And MAP kinase pathway is a very important pathway for multiple processes in the cells, including differentiation, proliferation, organ growth, but also cancer when, with somatic mutations. And a number of genetic diseases, like that I listed here, and there are more, belong to this family of diseases. And what's interesting for this, though, is that despite the gain of function in this pathway, you can have a very diverse outcomes on cardiomyocytes. So interestingly, those can be almost opposite. So for example, on the left, one of these mutations in this pathway that again give a gain of function result in cardiomyocytes become larger. So this is basically what we call classically pathological hypertrophy, so increase in the cell size. And on the right, in some of the other mutations, you can see that just if we look at this panel E, there is a number of cardiomyocytes in the heart, this is in humans, is actually increased. So some of these mutations result in enlarged hearts, but when you look, some of them are because heart has more myocytes, and another is because heart has larger myocytes. And sure, importantly, it will be interesting to understand the mechanisms of this so that when you go after therapy, you'll be able to address it differently depending on which type of a phenotype is present. Now, the pathway itself, as I said, MAP kinase or MAP kinase ERK pathway is extremely complex. It's starting from receptor level down to effectors. And the main effector in the pathway is ERK, so the kinase that is basically works both in cytoplasm and also when translocated in nucleus, induces a lot of transcription of different factors. And there are, in cancer, there are mutations that when they happen in between the receptor level and ERK itself, result in gain of function. So a lot of this pathway and the gain of function in these pathways was much more studied in the context of cancer than in the context of cardiac disease. But we'll use some of the tools from the cancer biology to try to look in what happens in cardiomyocytes. So specifically, just from that big complex pathway, what we've done is actually to overexpress and amplify this pathway by using an oncogenic mutation in BRAF, which is one of the kinases in the pathway. And if we overexpress it, we are able to, under the cardiomyocyte specific promoter MHCK7, we can just augment this pathway specifically in cardiomyocytes. As a control, we would just use lentivirus that only contains mCherry, and we'll be able to compare this phenotype. And so some of this work has been published. So I'll just try briefly to kind of show some things and how they relate to in vivo. So first, the idea is that if you apply, constitutively active BRAF or this mutant that I described, V600E, that you'll get ERK pathway amplification. And so if you just look in Westerns for ERK and phosphorylated ERK, you can see they are, significantly upregulated compared to the use of control virus. So we have a, continuous upregulation of ERK, and as a consequence, if I look in these cardio bundles and I look at cross sections, I can see that, with a constitutive active BRAF, there is an increase literally in size or larger size of this tissue. There is also, if you look EDU, much more of EDU incorporation in cells, so cells show more proliferative phenotype. And I don't know if it's seen on this slide, but if you compare on the bottom, left and right, the cross-striated phenotype that's present normally in the left is actually almost lost, and there is a sarcomer disassembly that happens with overexpression of this mutant. If you measure function of these cardiomyocytes and EDU incorporation quantitatively, you can see that there is an increase in proliferation, and then there is a decrease in tissue function. In this case, I'm just showing the force of contraction. And interestingly enough, there is no really hypertrophy. When you look at the cell size, you're not seeing hypertrophic phenotype, you're seeing more what's here as a pro-proliferative phenotype. If you look in some other markers of function, which is electrical function, in this case connexin 43 as a gap junctional protein, you can see also decrease of gap junctional again after 1 and 2 weeks of culture with a constitutive BRAF. And if you look at the electrical propagation, electrical activity using optical mapping, you can see that again there is a significant decrease in conduction velocity. So electrical spread across tissue is slower. So both electrical function and mechanical function are compromised while the cell cycling is increased. you can look at more on a whole transcriptomic level, and I'll not go much into details, but there is a really broad transcriptomic changes with constitutive BRAF expression, activation of this pathway. Thousands of genes are differentially changed, both after 1 and 2 weeks. It happens relatively quick. And when we validate some of this, we find that in these tissues, there's an increased glycolysis compared to the more of oxidative metabolism, which would be more of what you expect. There is an increased migratory capacity of these cells. And if you look all together, basically in this signature, transcriptomic signature, it shows a dedifferentiation phenotype. So cardiomyocytes here show something that looks more like a dedifferentiation and loss of function with that. So seeing this phenotype, we were interested to see what would happen in vivo. And the reason, because there is a possibility that if this phenotype would be pro-proliferative in vivo, that a lot of the gene therapies that are kind of now targeting cardiomyocyte proliferation could potentially, this would be a way to increase a number of cardiomyocytes in the heart. So what we've done, we've basically, just generated AAV vectors, so adeno-associated viral vectors, and express constitutive active BRAF, specifically in cardiomyocytes under the cardiac troponin T promoter, or in just a control M-cherry virus. And what we've looked at those, and this is what I'm showing here, is basically a cross-section of the heart, so septum, left ventricle, right ventricle, and EDU incorporation. And when you look and compare to the control virus, is a huge increase in EDU incorporation, and this is just quantified on the right. And so this was really interesting to see, and we were very excited that this could be potentially cardiomyocytes as what we've seen in vitro. But when we looked more carefully in where this EDU is incorporated, we realized it's actually outside of the cardiomyocytes. So cardiomyocytes here shown in F-actin, and all of the EDU incorporated is mostly in the vimentin positive cells. And so virtually basically all of it. And so what they told us that this is a similar thing that I didn't mention, but we've seen also in vitro, where we saw that actually there is also cardiomyocyte proliferation, but there is also surrounding fibroblast layer that also got incorporated EDU. But here there's no cardiomyocyte proliferation. There's no also increase in cell size, but what we've noticed is if you look at the cardiomyocytes on the right that got M-cherry, so these are the ones that got a constitutive active BRAF, and you look at the the sarcomeric structure now in this heart, you can see that there is a disassembly of a sarcomeric structure. So this results also in functional loss. So if you look at with echocardiography in function, you see that there is a decrease in function. What we haven't noticed, though, there is no increase in hypertrophy. So unlike in vitro, in what we see in vivo in adult hearts is basically no change in the cell size, no proliferative signature, but some of the sarcomere disassembly that we've seen also in vitro, as well as some of the fibroblast proliferation, but much more in vivo than what we've seen in vitro. Okay. So knowing that these diseases that are including, like in this MAP-ERK pathway have an early kind of a phenotype, we've also wanted to see what would happen in the environment that's more permissive to proliferation, which are neonatal rats. So what we've done, we've injected AAV vector, but this time we put it in a P2, so postnatal day 2 rats, injected the virus and checked what happens 7 days later. And so in this case, when you look at the cross sections of staining, and you stain with the WGA with germ agglutinin, which kind of shows the boundaries of the cells, you can basically quantify that and see that cross-sectional area, if you look in the individual cardiomyocytes, is significantly increased. Not if you look across hearts, but if you look across cardiomyocytes. And to better assess this phenotype, we actually looked in isolated cardiomyocytes from these P9 hearts, and then evaluated them across many cardiomyocytes upon isolation. And what you can see here that, interestingly, we noticed that there is a hypertrophy in this case, but it differs in a sex-dependent manner. So in males, as you can see here, there is actually a significant increase in width of cardiomyocytes, something that we call concentric hypertrophy, while in females, there is a significant increase in, in females, there is a width which is concentric, and males, there is a length which we call eccentric. These are two different types of hypertrophy, and interestingly enough, in humans, this is exactly what's seen. So for now, unknown reasons, concentric hypertrophy happens much more frequently in females than in males, and eccentric hypertrophy, which is more deteriorating actually for heart function, happens in males. So it's interesting, mechanisms of this are not clear, but one, interesting thing, and there is a functional decline also here. And again, I'm not showing echo data, I don't want to, show too much, but one interesting thing is that if you look, first let me show you, if you look here at the ERK activation is not different between males and females. So it seems that they are relatively comparable in terms of how much the pathway is activated. But if you look at the survival curve, basically what you see that within 10 days, almost all males die, and while females continue to survive. So, again, why this is happening? We are basically studying this now. So it's interesting that we've tried to do this also from neonatal rat isolations in vitro and separate them in females and males and try this, and we have not seen any differences, sex-dependent differences. But in vivo we see these differences. So our working hypothesis that we are working now is basically that there are actions. Because we see the same activation of ERK pathway both in females and males, there is something that's sex-dependent, that's non-canonical. So basically not like a classical kinase pathway, but more of a kinase-independent that is differently regulated in females and males and can induce different type of hypertrophy, eccentric or concentric. Okay. So at this point, we are working a lot on trying different mutations that are present in humans, using human iPS-derived cardiomyocytes, and specifically trying to control as much as possible this pathway to be able to see, can we reproduce in vitro concentric versus eccentric hypertrophy, as well as hypertrophy versus hyperplasty. All right. So second topic I want to talk about, in terms of engineered heart tissues is basically, our recent effort to make them more realistic, or, as realistic as you can. And, one way to do that is that, realize that, while we have, and many others, have initially developed cardiac engineered heart tissues for, a lot of in vitro studies, one of the important components that's usually not present is actually the presence of autonomic nervous system. So autonomic innervation has a lot of physiological effects, but also what we call tropic effects. So on the left is, classical, effects of sympathetic innervation in the heart, which is this increased heart rate with a sympathetic tone. Inotropy is contractile force, lusotropy, how fast heart relax, and so on and so forth. On the other hand, sympathetic innervation has been shown to have effects that are not in the classical physiological sense, but rather they increases cardiomyocyte maturation. It's basically some little evidence on that, decreases potentially proliferation, although there is, again, contradictory evidence on that too. And it's also something that's interesting, that it's really seems to be required for regeneration in younger hearts. On the other hand, in older hearts, if you have sympathetic innervation, often is something that after the infarct, for example, there's a aberrant sympathetic ingrowth in an injury site. And due to that, there is a arrhythmogenic activity that's dictated by overactive sympathetic neurons. So it would be interesting if one can make tissues which are innervated functionally with autonomic nerves and then use them to study these types of phenomena, either physiology or injury response. Okay, so to be able to do that, we've differentiated sympathetic neurons by modifying some of the methods that have been already developed and using this line of iPS cells that has this EGFP reporter in the endogenous locus of FOX2B, which is just neuronal transcription factor. And based on that, we can kind of separate or sort out cells that are going into the sympathetic neuronal fate. And after few weeks of differentiation, we can show that these cells start to form basically clusters of neurons and neural outgrowths, and this is shown at day 30, and these outgrowths become much stronger and more abundant by day 50. You can look at the FOXO2B expression that with maturation increases in these cells, and also if you look at some of the enzymes that are characteristic of sympathetic or norepinephrine secreting cells like tyrosine hydroxylase, TH. That's something that's present in sympathetic neurons, but not parasympathetic neurons, for example, or cholinergic neurons. It goes up, while some of the cholinergic enzymes like CHAT, choline acetyltransferase, are low and decrease with time. So basically, this shows, and with other studies that we've done, that these sympathetic neurons are really having sympathetic fate and secrete norepinephrine, which is functional. One way to test for this functionality is also to look at the response to depolarization of the membrane. So if you add potassium chloride or nicotine, you should be able to see calcium spikes inside these neurons. And so if you perform recording of calcium using the calcium indicator, either dyes or genetic sensors of calcium like GCAM, you can see that with addition of nicotine, there is an increase in calcium transient and response to nicotine, which we've also measured by ELISA, induces increased response in norepinephrine. Okay, so while neurons are functional, the question is, can we generate these innervated heart tissues? And one way to approach this using the same frame gel technology is to generate two compartments. One which will be containing the IPS sympathetic neurons on top in green, and another one is our classical cardio bundle. And if you do this approach, you can co-culture these two cell types in hydrogel and see whether with time there is an innervation of a cardiac compartment with ingrown neurons. What's important about this system is that the sympathetic somas, right, cell bodies of sympathetic neurons are separated from heart, which is how the sympathetic innervation is separated anatomically. So you are basically separating somas. With that, if there is an injury in heart, there is no injury to neuronal somas. And this is something that mixed co-cultures do not have, cannot mimic that important part of the anatomy and physiology. All right. So if we do this and we look, what happens after 4 weeks in culture. You can see that there is an ingrowth of neurons only in the cardio bundles that are co-cultured with sympathetic neurons, and you can see them stain for neurofilament heavy, just one of the neuronal markers. And this, happens throughout the kind of, the length of the tissue, as you can see here. And if you look more in detail, you can see the presence of neurons and also the presence of these little, thickening parts, which are called synaptic varicosities, which are the sites where supposedly, although still not proven, there may be a neurocardiac junctions forming. Of course, we don't have a proof for that, and nobody has it, but there is a one way that, within the heart, this presence of this, sites also labeled with synaptin 1 here, synapsin 1, in white, is actually where that communication between the sympathetic neurons and heart cells may happen. If you look at the size of these neurons, it's really small. So basically less than 1.52 microns in diameter, which tells you that these fibers are likely non-myelinated, similar to as they are non-myelinated inside the heart. Okay. All right, so we've looked whether the presence of neurons and our co-culture media, do they change functional properties, and we do not see any change in force generation, active force, passive force, specific force. We record action potential propagation, do not see any changes there. So we're able basically to incorporate these sympathetic or autonomic neurons without, compromising the baseline function of these tissues. Now, what happens if we, look at the spontaneous beating? And it's understood and known from physiology that presence of sympathetic neurons actually is chronotropic, as I mentioned, so it increases spontaneous beating rate, but also increases variability of the heart rate. And healthy heart has actually healthy dose of variability in the heart rate, right? So presence of sympathetic neurons does both, in our case, both increases beat rate, spontaneous beat rate, as well as the variability of this rate, as expected, And this happens because of small spontaneous releases of norepinephrine from the sympathetic neurons. And if you apply nicotine to specifically activate, sympathetic neurons, you can see that in the, the cardiomyocyte only, so cardio bundles, there's really no increase in rate when you add nicotine, but when you add nicotine in the, in the innervated cardio bundles, you see expected increase in rate. And so, this is also, just quantified here. The bottom line for us is that we are planning to use this along with the next model I just quickly showing a couple slides to study the injury response. So what we are doing, we have developed a model of cryo-injury where we locally injure these tissues, and we can look in a re-innervation and effects on arrhythmias and cardiac function. Of course, like this, we also try to do this because we've published and shown how we can incorporate vasculature in this. It's non-perfused, but using iPS-derived endothelial cells. And so we want to develop conditions for 3 culture, being able to 3 culture all these tissues. And then the idea is here that similar to how the development happens, we have cardiomyocytes and endothelial cells mixed in one compartment, and then neuronal compartment and innervation happening of sympathetic neurons into the vascularized cardiac tissues. So now, when you look at this and study specifically for neurons or endothelial cells, you can see here in the 3 culture presence of all cell types. And if you look at the, whether the 3 culture affects things differently in terms of cell abundance compared to 2 culture, you see no difference. So, for example, on the left, you have a same number of endothelial cells present, whether it's innervated or non-innervated bundle, right? And on the right, it's similar thing for neurons, whether they're vascularized bundles or not. There's a similar level of, of the neuronal area. So, and functionally they remain the same. And if you look at some of the, again, responsiveness to nicotine as a way to test functionality of this innervation, you can see that basically as these 2 little stars will here show, that response to nicotine is only present when you have neurons, be it with endothelial cells or without endothelial cells, and there's an increase in bit rate. Okay, so in summary, for cardiac part that I want to show, we you use both rodent and human cardiomyocytes within this cardio bundle model or cardiac tissue patch or cardiac networks, which I didn't have time to talk about. We are interested in studying rare genetic diseases, specifically for MAP kinase pathway and neuropathies, because they really give these diverse outcomes that nobody understands where they're coming from. And then, we are interested, of course, in building more realistic test beds for drug testing, disease modeling, where we include other cell types. We include vascular, neuronal. I didn't have time to show immune system cells, and then or fibroblasts, and then are looking into the injury response in these models. Okay. All right. My second part of the talk, I'll tell you a little bit about our engineered muscle. So it's basically the same idea. It's the same system where we start to make these cardiac bundles, but we now, because these are made of skeletal muscle cells, we call them myo bundles. The idea is to, again, mix cells and hydrogel, put inside these molds, have a frame around that will anchor the gel. That anchoring of a gel eventually results in the formation of cardiac bundles. And if you are basically trying to do that from primary human cells, just a little needle biopsy of 50 milligrams from a muscle, like say deltoid muscle or some other muscle in our body, vastus lateralis, and so on and so forth, you can get about 1,000 of these myobundles. So it's a relatively system that's scalable, even with primary and very small biopsies, right? But we get biopsies, we get also surgical discards from primary cells, and of course we've developed iPS technology to be able to do it. After a couple of weeks in culture, when you look at this again, histologically or you do immunostaining, you can see that these very much look like muscle fascicles where muscle fibers are aligned, and then there is a layer of fibroblasts again surrounding these muscle layers. In a cross section, you can see all muscle fibers basically being filling up this space. And if you just blow up or zoom out this image, you can see that and stain for example, dystrophin or spectrin, the membrane proteins that are present in mature fibers, muscle fibers, you can see them nicely kind of, delineating individual muscle fibers. And we can use this as it's used in native muscle to study muscle fiber size, hypertrophy, responses to injury, and so on and so forth. All right. One of the important things is for muscle, unlike heart, is that it's highly regenerative. So one of the, the ability for muscle to regenerate is due to the presence of stem cells that reside in the muscle. They are called satellite cells. And so within these engineered muscles that we make, they also have this regenerative component. So there is a satellite cell niche that forms, and satellite cells, like shown here, that are stained with this transcription factor PAX7 are present in the muscle. They basically form in between the niche, in between the muscle fibers, and through M-cadherin, the junction, the cell-cell junction, they are basically connected to muscle fibers, and this keeps them to be quiescent, non-proliferative, and upon injury, they can proliferate and regenerate the muscle. And we've looked, using single-cell transcriptomic in this population specifically, and interestingly enough, within our engineered muscles, they basically, cluster into two clusters, the PAX7 cells that are that are more quiescent or more activated based on their transcriptomic signatures. And so this is exactly what you see in actually human native muscle. When you isolate the cells, there is a more primed population, and there is a population that's deeply quiescent, and a lot of the same factors shown here in this system are actually present. Well, so while this system is, of course, this is all in terms of, showing in terms of, the molecular makeup and histological appearance, what is most important, of course, is function. So we've shown the first time now more than 10 years ago with primary cells, and then later, 3 years later, in iPS cardiomyocytes, that we can generate this muscle to be functional. If you stimulate them once, you generate a twitch. And if you stimulate them with a higher frequency, these twitches fuse and form a tetanic contraction, which is classically how our muscle contract. So we use this system to study different things, and I just kind of give you snippets of that before I go and just describe at the end of disease modeling, which is what we primarily have this intended to do. So we use this to study exercise. In particular, we could apply as a chronic electrical stimulation. So what you see here, 2 carbon electrodes shown here as as these like these graphite electrodes, right? And they stimulate the muscles, and you can see that they are anchored on one side, and each stimulus there is a contraction. And we can use this to basically impose different types of exercise. So we can exercise for 1 hour, stimulating, then we have a rest. We can do this 3 times a day, or any times of day. We can do something that's more like a resistance training or more like endurance training, and use that as a muscle. And just to show as a example of one of a couple of these regimes, so this 1 hertz and 10 hertz, you can see that there is a functional improvement. So basically, these muscles bulk up, similar to if I'm to go to gym. There is a hypertrophy of a muscle itself. There is improvement in muscle function. And there is, improvement in metabolic flux and many other parameters that we studied, that happen like in a normal exercise response, right? We looked some in inflammation, and especially how exercise can counter inflammation to induce, inflammation type of kind of stimulus. We applied interferon gamma, a typical pro-inflammatory cytokine, and we looked, saw that histologically the muscle under interferon gamma actually deteriorates, but with exercise, it is actually recovered. A lot of this function is recovered, and then we've studied the mechanism of this, and the mechanism is really like direct activation of JAK-STAT pathway. So usually activation of JAK-STAT pathway happens with the pro-inflammatory cytokines. Exercise directly counteracts this pathway, and then JAK-STAT inhibitors counter fully this pathway and can fully recover what we've seen here. So so again, this is the way to show that we can, apply this to study classical physiology or, inflammation as a classical kind of pathological insight. Now, similar to cardiomyocytes, which I didn't show, we've also looked recently in vascularization of these muscle bundles. So basically, we would apply endothelial progenitor cells. These are human endothelial progenitor cells derived from umbilical cord, blood, and then a primary human myoblasts. And we would mix them and look, and this is a mixed culture. The main thing is to develop a co-culture media, which is not a trivial mixing of just muscle and endothelial cells, but one way or the other, after we develop this human plasma-like media, we've been able to co-culture this for a longer time and show that, there is a presence of vascular cells. They co-aligned with muscle. You can see them CD31 staining, or M-cherry labeling. If you label them with lentivirus, you can see them in cross sections. Some of them start forming lumens, and this number of lumens increases with time, which suggests that they're potentially maturing the endothelial cells. Functionally, there is no difference at all between a pure muscle or muscle plus or vascularized muscle cells. So from multiple aspects, again, that we studied, we see that there is no difference. But where there is a difference, which is interesting, and what is shown kind of in vivo, is basically if you look at satellite cells, so muscle stem cells, you can see that they're co-localizing with endothelial cells. And this, so they're both co-localizing, which is, quantified here, for example, in, Pax density closer or to the capillaries or further from the capillaries, and not only did they co-localize, but also the amount of proliferative marker Ki67 is lower, and some other markers, so which tells you that these cells are in a more quiescent phenotype and closer to the endothelial cells, are closer to these capillary-like structures. In vivo, this is the case. So muscle satellite cell niche actually does not only contain myofibers that I've shown before, but also endothelial cells and neurons to some extent are contributing to the quiescence of the niche and proximity of satellite cells to the vessels. So this is kind of something that you also see in vivo. All right, I'll finish with the quick examples of muscle, how the modeling of muscle disease, which is why we developed this system. So one is Pompe disease, and Pompe disease is basically deficiency in the enzyme called alpha-acid glucosidase, GAA, and either that enzyme can be completely missing, which results in a very severe disease phenotype called infantile onset Pompe disease, or it can partially be dysfunctional or missing, in which case we have what's called late onset Pompe disease, which is less severe. So we were interested to model the most severe form, which is infantile onset disease, and we're able to do that by basically finding, luckily being at Duke, which is one of the first sites that has developed current therapies for this disease, is where we could get a muscle biopsy from really infants actually to be able to do this. So anyway, so we've looked at the multiple donors, healthy and infantile onset donors. We made the muscle bundles, and then we looked in their phenotype, and one of the major phenotypic features or histological features of this disease is a lysosomal accumulation, where glycogen accumulates in lysosomes, and you can see here by staining with the lyso tracker, there is actually increase in infantile onset myobundles compared to healthy, significant increase in lysosomal in lysosomes. Also, if you look at some of the proteins like LAMP, lysosome-associated membrane protein, is also upregulated. Again, classical histological hallmark of this disease. Biochemically, if you look, you can see that enzyme activity in the infantile onset is, biomodules is gone as expected, and glycogen, which is, this is a glycogen storage disease. Glycogen is accumulated over, a couple weeks in this infantile onset myobundles, which is again a histological hallmark of the disease. Now, interestingly enough, when we wanted to look functionally what happens, which is muscle weakness, we did not see it really at the baseline. And this is maybe because this is after 2 weeks or even after 3-4 weeks. It's maybe the muscle fibers are immature, maybe the whole process is, takes time to develop, or glycogen accumulation takes time to affect function. But one way for us to be able to tease out this functional deficit was basically to, apply, drugs that would suppress actually, another type how glycogen is processed in the cell. So with that, we basically only make cells to be able to process glycogen through lysosomes, and in the infantile onset Pompe disease, that pathway is gone. So basically, we leave them without at all possibility to process glycogen. And with that, now we can precipitate functional phenotype. And if you look at fatigue test, which is shown here, which is you basically keep stimulating, this muscle to contract for 30 seconds. And of course, it will fatigue over time. You'll see the force dropping. You can see that there's much more fatigue in the infantile onset patient disease. So again, this is primary cells, applying them in the disease modeling, but really seeing functional aspects of the disease only after certain challenges, like drug challenges in this case. All right, something like another type of muscle dystrophy, and is the one that Duchenne muscular dystrophy is much more well-known and much more prevalent in boys with Duchenne disease, where dystrophin is lacking. And what you have is a lot of muscle injury, degeneration, regeneration, and unfortunately, these kids live not more than early 30s at this point. So what we were again able to find is primary muscle biopsies from 2 patients. These are very hard to find because the muscle is already wasted, and we were just lucky that they underwent scoliosis surgery, which is often what happens, and we were able to get some discarded muscle to and then have a healthy lines for comparison. And we would basically again make Maya bundles and look in them. Now, one of the important things that I want to mention for this disease, and it's not really modeled, and you'll see, by any other systems, is the presence of senescence of cells that happens in late stages of diseases. So usually these boys go into wheelchairs in about between 10 and 12, and with that in particular, but always as they are approaching that, their muscle stem cells become senescent, and their tissue cannot regenerate, which is when their weakness and immobility happens. But that's not been modeled yet in a dish, basically, right? So what we've looked, we analyzed these primary cells, compared them to healthy cells. We found that they have a less proliferative capacity, they are larger, and then we did a really comprehensive studies to basically look at whether they are senescence. And we've compared them with all other ways that how we make muscle progenitors. So we compare them either with the immortalized muscle progenitor lines, these are immortalized lines from muscle cells, or we compare them with a small molecule differentiation of iPS cells, or with a reprogramming differentiation of iPS cells, which is this PAC7 overexpression model that we usually use also in our lab. And none of them actually showed any features of senescence, but the primary cells. So primary muscle cells from biopsies here really are showing features of senescence here, like proliferative capacity decrease by all of these other markers that we've looked at that they shown, including mitochondrial dysfunction and ROS formation, P21 and 16, and so on and so forth. All right, so using these cells now, which are senescent in origin, we made bundles. And here is how they look in the cross-section. So healthy, as you've seen before, form nice dense muscle fibers, but DMD actually have smaller, more variable myofiber diameter and much less myofibers, so they are able to form myofibers much less, which is expected from senescent cells. Now, what you see, and I think first time here, is really clear phenotypes that are found in vivo, which is one of them is shown on left. That's basically a muscle branching. So fibers in DMD are branched, and also hypercontracture, which is when fibers basically detach from the matrix, not having dystrophin, and they basically ball up. So you can see them inside here. And if you look at other parameters like metabolic function, their oxidative metabolism is decreased. Their mitochondria are functional. This is here, and also their ROS reactive oxygen species are increased. Their function is severely decreased, and you can see here with using different parameters. Red is DMD, function is specific force generated per muscle area is really decreased, and then muscle fatigue is increased, so they are fatiguing more. So all these features basically have shown also in vivo. And then finally, we've developed this assay, which is well-known assay, in which it shows the vulnerability of muscle membrane to eccentric exercise. So when you perform eccentric exercise, it's specifically when the muscles of Duchenne boys are vulnerable to, and you can see that in the force loss that's kind of shown here is much more increased after this eccentric injury in DMD muscle. So this model is a model of advanced DMD, with a senescence, which has not been shown before. And we use it to actually look at some drugs, and that, we've shown that basically, if you apply all these kinds of different drugs, and some of them are clinically used glucocorticoids, others are experimental types of drugs. And, as you can see, none of them really improves contractile force compared to the, original muscle. So none of them have real functional benefit in terms of improving strength of contraction. But vamorolone among these is the one that actually has, protects muscle from eccentric contraction. So vamorolone is just newly approved type of a steroidal drug, and is a alternative to classical glucocorticoids such as prednisone, and all of these boys are on. So basically, that's an interesting kind of finding that so anyway, so this system now serves to us to study more mechanisms of this senescence and maybe ways to try to prevent or counter it. That we are also studying in mice now in the model of the Duchenne, D2 model in mice. Okay, and now I'm coming close to the end, and I want to just to mention another disease because it's interesting from a point of view that it's also dystrophy. It's a little bit more rare than than Duchenne muscular dystrophy, but it's really characterized with muscle weakness, immobility in later ages, about 30, 40 years of age. People end up usually in wheelchairs. But one interesting phenomenon there is that basically their muscles can be fully replaced with fat. So unlike in DMD where there's a lot of fibrosis, there is there is no fibrosis in dysphilonopathy, but there is a complete replacement of muscle with fat. And mechanisms of that are not known. There are no clinical trials, no curative therapies for these patients. And so we've basically first time developed a model using iPS lines this time, obtained from Jane Foundation that is specifically interested in this disease, and we were able to compare the phenotype and show first time develop a disease model for this and study mechanisms. And what we've done first, we've generated muscle bundles again from iPS cells, 3 lines shown here with this disease LGMD2B and healthy counterparts, and structurally there is no changes. So structurally the muscle fibers look great, and the same fusibility, same myogenesis, same cross striations, structurally everything looks good, same fusion capacity, but when you go and look at function is severely compromised. So basically it's about 50% of force generation, and that's something that you see also in calcium. And if you look at other things which is a vulnerability to injury in this system, which is shown here, you can apply a hyposmotic shock, so low osmolality media, and muscle membranes tend to blow. And with this disease, they're much more vulnerable to these forces due to the swelling, and they die. So basically, you can see how muscle fibers are all destroyed in LGMD2B, while they remain relatively intact in healthy muscles, and completely destroyed actually with this in LGMD2B muscle. So this model actually is a model that you can study injury responses, and then drug action and so on and so forth. Another property of this disease is metabolic dysfunction. So if you give these muscle fibers fatty acids, they don't react to it. Basically, they don't increase their oxidative metabolism, which is very normal for normal muscle fibers. So there is also mitochondrial dysfunction. Again, just shown with different mechanisms. So it gives us a really good model of muscle that a lot of the hallmarks of disease are reproduced in this model. All right. So we looked a lot on this from multiple points of view, transcriptomic, metabolomic analysis, and I just want to show you one thing that we found that we are now testing in vivo, which is that cholesterol metabolism in these tissues is really, very much dysfunctional. And in particular, there is a significant increase in free cholesterol, even in the absence of exogenous cholesterol. So you can have a media without any cholesterol, but muscle itself produces much more free cholesterol, shown here by this Philippine stain. And also, when you look more in more detailed, products of metabolism, you can see that specifically cholesterol esters, shown here as CEs, and then all these cholesterol ester species are actually significantly upregulated in this muscle. So after a lot of understanding, doing metabolomic analysis and trying to understand what's wrong, we realized that cholesterol synthesis is compromised, and also cholesterol trafficking to the membrane. And if we use drugs that are some of them clinically available for other diseases, to basically either statins, which are classic drugs against cholesterol production, shown here, or this drug that inhibits this enzyme in a cholesterol ester production. And if we looked in basically functional effects or response to injury, they were all improved significantly compared to to the vehicle control. So what we're doing now, there are mice. It's not a great system. They're called bludge mice, and we are basically, for 6-month-long experiments, we are injecting these drugs to see whether they will revert the phenotype that's present in these mice, in specific muscles. All right. To summarize this, and this is my last slide, we used human myobundle studies for both primary and iPS-derived muscle cells. We measured different types of physiological, drug, and injury responses, which allow us to model disease and to understand the mechanisms of disease, which we use we apply for, at this point, Pompe, Duchenne muscular dystrophy, and dysphalanopathy. A lot of the focus has been in dysphalanopathy because of its complexity, and it's a very scientifically actually interesting disease. And then we are incorporating, similar to cardiac bundles, different cell types to be able to better model these diseases. And especially actually in dysphalanopathy, I mentioned fat, and it's really dependent on the presence of immune cells and muscle interstitial cells. So although I didn't show, we've been the first time able to show fat formation, ectopic fat, in this model when we incorporated macrophages and interstitial muscle cells from patients. Okay. Thank you very much for listening. I know it was a lot of things that I talked about. These are the people who really did the work, and our collaborators. There are many of them, both in heart and muscle. Thank you. I'll be happy to answer any questions.
Thank you. It's a very interesting talk, and then it's overlapped to my previous work when I was a postdoc. I'm talking about 6 years ago. So back then, I was trying to set up some NMJ co-culture, motor neuron coming from the HB9, report line, and PAX7, the report line drive the muscle progenitor cell, and then I tried to set up the co-culture for the NMJ. And then neuron side, I tried to use the high throughput functional readout using the MEA, but I couldn't figure out the best high throughput quantification method for muscle side, like twitching quantification. Oh. But it seems like you did for the drug screening. So how much they are efficient and
well, we use mostly genetic indicators of calcium, and that's one of the imaging readout. It's not. This system is not very high throughput, right? But it's kind of a medium throughput. We can maybe do 96 of this at a time. So we cannot do thousands. Yeah. So, but most of our readouts rely either on dedicated functional measurements like force. Yeah. Or imaging readouts, which are amenable to higher throughput, yeah, and that's mostly imaging calcium. Okay. So we've also done NMJ cultures. I didn't have time to talk about this, and again, in a similar two-compartment model as I've shown for heart, and there is of course functional innovation. I just showed heart because heart is one of the rare systems that this still exists. NMJ systems are more frequent. I think important for NMJ systems that has not been shown yet, and we now are kind of preparing to submit, is that nobody's shown really the most important part in NMJ cultures, which is that you can stimulate nerves and induce tetanic contraction in muscle. That's called tetanic transmission. Usually, that's the hallmark of functionality, and so far people have never been able to show that. We see that, luckily.
So. Thank you. So when I remember the DMD, IPS line, it has a lot of, differentiation issue using the PAX7 report line. So is there any chance to look at the isogenic control to see any phenocopy differences between disease line and isogenic control?
Sure. So now, for example, for dysphalanopathy, we've done this the phenotype was very consistent between the healthy versus diseased lines, the 3 healthy and 3 diseased lines, but they are not isogenic. One of them is a heterozygous sibling, which is just a carrier. So at least there are 2, basically twin brothers, but one had another somatic mutation that protected them. So anyway, but we are now, used CRISPR-Cas9 technologies, and we've just knocked out dystrophin completely. So, so we have these lines to compare also in isogenic setting. How consistent and reproducible this is, I don't think that we'll see anything different, but it's a good control, of course, to have.
Thank you. Yep.
You spoke during the first half of your talk about sort of an interesting phenomenon, which is the differentiation in a male phenotype versus a female phenotype in this hypertrophy. I'm really curious, have you seen yet, and I'm not sure if you've done these studies, but how much of that seems to be related to intrinsic factors versus extrinsic factors? So basically, the source of the host, the genetic sex of the host, of the source material versus, for example, the media components adding hormones and things like that. Do you have any thoughts?
So what I've shown as a sex dependent was all in vivo. So it was, but it was in young animals. So I don't think that really hormonally it's as important because I think they are pre-puberty, if you will. So so I think this is something to do really most at linked to X chromosome or rather than really hormonal. Although but we haven't studied it in detail. So we are now gearing to do some of the proteomics and trying to understand the differences. These are not easy studies, and I think you probably know. So but it's a very very interesting and remarkable to see how clear is how definite was the difference in survival. So we really didn't expect that. It was just like when mice start to die, and the rats start to die, right? Just kind of almost accidentally, we realize they're all actually males, and then so we'll see where that takes us. Yeah.
Thank you, Nenad, for an excellent talk. It's really inspiring, especially the detailed characterization and everything. So my first question is the innervated myobundles you have developed. What was the experimental setups for that one? Because you have shown there is a neural cells and the myotubes formation, and it is coming, the innervation is coming. So what was the experimental setup? How difficult it was? Because are you seeding the cells at the same time, or how difficult it is to create those kind of things?
So luckily, the way how the compartmentalization is set up, right, with this frame that has basically the frame acts as a compartment. The frame goes into mold, and basically there are trenches under the frame. And so once you put the hydrogels with neurons in one compartment and give it maybe 10 minutes, you can then completely independently mold the muscle compartment. Okay. And the way how depending on the amount of media or cell gel amount and depth of these trenches, you don't have any spillover into the other compartment. And so basically the two form like neurons and the muscle, and then the rest is self-assembly. So basically neurons it takes time, about 2 weeks, but then they'll start to grow into the into the muscle. Muscle fibers, on the other hand, they just align and form myotubes, similar to what they would always do. Okay. So so it's done basically simultaneously, right, within 15 minutes. Okay. Between the two compartments.
The second question is like you have exactly mimicking at most like 98%, 99% of the anisotropy of muscles and everything. Have you ever thought about replacing, for example, volumetric muscle loss treatment? Have you ever used your like a fabricated graft for muscle repair in animal studies?
Yeah, sure. So we've shown before that we were able to implant this in a type of a volumetric muscle loss kind of model. They survived, they vascularized, there is a fibrosis forming, but these were also human in rats. So they were in mice. They were like immunocompromised, nude mice. I don't know if they're fully like preventing rejection, but we did not go much more into that. For us, we just demonstrated they survive and that they function through calcium imaging, but we haven't done much on like a detailed assessment of muscle function recovery, potential innervation, and things like that. Innervation, of course, is one of the biggest issues in the whole this, because as far as I've seen so far, there's really no rigorous demonstration that implanted muscle tissue in a volumetric muscle loss ever innervates from a host. There are some anecdotal evidence, but not a strict experiments.
Okay, thank you. Appreciate it.
Thank you for sharing your great work. I'm really interested in your co-culture platform. So did you ever try to co-culture with the immune cells and macrophage to explore the immune injury or immune therapy or immune reaction?
Yeah. So we've done that in two contexts, but I think the one that's really interesting is the one with with the within the dysphalanopathy context.
Yeah.
So I've shown a lot of the features of disease that are replicated when you only have muscle, right? Like from a point of muscle itself, that's almost muscle autonomous. But in this disease, it's known that incorporation of macrophages and abundance of especially like M1-like phenotype is actually even maybe precedes the onset of disease. This disease is a late onset, happens about 17, 18, 19 years, starts to show in humans, although they could be world-class athletes before that, and also shows up only in certain muscles, but not in the others, which again, no one understands why late, why in some muscles, but not the others. One way or the other, without incorporating macrophages or muscle interstitial cells, which are what we call fibroadipogenic progenitors, there is, in this model, there's no fat formation. But that's a hallmark of disease. However, when we incorporate both, and especially in an injury setting, we see first time that there is a, ectopic fat formation. So that part of the phenotype, the immune system cells seems to be very important. We believe and kind of have evidence for that for now, that immune cells alter the fibroadipogenic progenitors in their differentiation capacity and steer them towards going into fat rather than fibrosis. And that's why the fat formation.
Yeah, thank you. And I still have another question. A small question, yeah. In your different project, you choose the primary human cells and iPS cells. Why you choose different cells for the different project?
Well, the main reason is abundance of biopsies. So, for example, if we were to study dysphalanopathy, iPS cells are easily accessible from, blood draw, or, but muscle biopsy in people with a degenerative diseases of muscle, there are many types of biopsy just to establish their phenotype. And they're really not happy to give yet another biopsy, even for the research, right? So here and there, we're lucky to get some of these biopsies, but and because primary cells are still golden standard, we think. But but in absence of being able to get those biopsies, we go with iPS cells.
Yeah. Thank you so much.
I have a quick question. Very nice work. Do you know why heart muscle doesn't get cancer? Because you said some relationship.
It's funny you ask that. I don't know if there was a science paper just last week, maybe, or 2 weeks ago, that looked at that and basically, found that, the mechanical contractions of heart, and I don't know why that's not still the case in skeletal muscle where cancer is much, prone, but the mechanical contraction of cardiomyocytes seem to be activating Nesprin-2, which is a nuclear envelope protein in nucleus in cancer cells, and that basically downregulate proliferative signature in cancer cells. So they've done experiments, they've taken some human kind of samples, but they've done a lot of experiments where they would just implant cancer cells or inject them in the heart, and they would transplant the heart in the neck, which would mechanically unload the heart. Those heart with cancer cells in the neck would that mechanically unloaded, would develop and replace some of the heart cells with the cancer cells. Those in a beating heart would not. And this was, they've shown, through multiple studies, basically what happens transcriptomically in single cell and whatnot, but it comes down to mechanotransduction from heart cells towards muscle cells through this protein Nesprin-2 that they believe could be a therapeutic target now, although I'm not sure about that. But, that's a longer story, but that's the reason. It's funny you ask that because 2 weeks ago, I may not have much, but hand waving. This just happened, this paper just happened, recently. So
thank you. Other question?
The NMJ has a pretzel shape. Do you know why it doesn't form pretzel shape in vitro conditions?
We've seen only sporadically a pretzel shape in I didn't show any NMJs here for because I talked about cardiac neuronal, not muscle neuronal, right? But NMJs, of that more elaborate pretzel shape that they call, is we see only here and there in our studies, and we've kind of characterized that in terms of shape. It's not easy. There's that so-called lacunarity that you do and can characterize. With characters, and we've seen that lacunarity or complexity of these NMJs in our tissues increases with time of culture, which says they are more mature, but they're nowhere close, I think, to adult. So in vitro, I don't think that anybody was really able to show adult NMJ.
Thank you.
Great talk, of course. A couple questions. First is follow up on Alicia's question on that male female, right? So could you guys, you showed in the rat model, right?
Yeah.
Were you able to recapitulate that phenotype in vitro? Because if you can, right, of course you can titrate the, estrogen, testosterone,
Right,
and
sure.
to answer that question about intrinsic versus.
So the only thing that we've done in vitro, thinking that it's probably the most relevant, was to isolate neonatal rat cells, specifically from female pups and male pups, make the same, introduce the same mutant, BRAF, like a constitutive active BRAF, and see whether they would be functionally different. And we saw nothing. So we repeated that and saw nothing again. So I don't know that what we are missing from, in vivo environment, but, there could be then many other factors. So.
But let's say if you take the adult one that has already been infected, so they already have that phenotype, but then in vitro, if you alter like estrogen and and the testosterone, would that kind of?
Sure, it's a possibility. We haven't done those experiments to try hormonally to see, whether that would make a difference. Yeah.
Second question is about that, second part, the sympathetic nerve innervating. First, just very quickly, it's so fast. We study parasympathetic, right,
Yes.
mainly, and go to. But can we use the same system? You think that would work? Like if you put in the chamber, let's say we want to study pancreas, liver, and we use your system, like I put a parasympathetic cell antibody there.
The only issue with that is as far as I know, there is still no, at least for human, good ways to differentiate parasympathetic neurons from iPS cells. There is we've made some progress. We've been more focused on sympathetic because it's a lower hanging fruit. For parasympathetic neurons, there's maybe one paper out there that, in our hands at least, we cannot reproduce what they say, but, we have alternative methods. Still, we are far from what we are able to do with sympathetic neurons. So that's the only culprit could be maybe if, otherwise in terms of compartmentalization, sure. And we also are making 3 compartment models, which I didn't mention, but if you want to apply sinoatrial nodal cells and cardiac cells and innervation, because a lot of innervation happens through the nodes in the heart, then there is a possibility to do it as a 3 compartment rather than 2 compartment model. One way or the other, yes, compartmentalization is possible. Parasympathetic neurons, I don't know about that.
We can potentially just take a adult primary, right? We can get from nodal ganglion and
Mouse?
Mouse.
Yes.
Yeah, humans
yes,
different
yes,
story.
yes. That's a possibility.
So the third question is the last one, the last disease that get replaced by fat, right?
Uh-huh.
So is that a germline mutation that the patient covers in all cells, or just happening somatic in?
Exactly. So dysferlin as a protein is very well expressed or mostly expressed in muscle and heart. However, it does express in macrophages and does express a bit in endothelial cells, at least. It's a little bit contradictory, but not many other types. So most of the phenotype is expected to be coming from cell autonomous and maybe immune system cell involvement. That said, it's very interesting, and we reproduce that also in our system. Heart has 0 phenotype, despite lacking dysferlin. So the canonical role of dysferlin in protecting the membrane from rupture is actually the same in muscle or in heart. When you don't have that protein in muscle, there's a huge phenotype. When you don't have it in heart is somehow protected. When we do this with iPS lines, we don't see any phenotype in iPS-derived cardiomyocytes. And we see all this phenotype in muscle. So somehow it reproduces in a dish. It's very interesting to understand why. And, we may be looking in that, but, so it's,
So I guess my question is, right, if you take that muscle, if you put I don't know if you can, if you derive macrophage from the same iPS, or you take macrophage from let's say different person. So my question is, does that mutation also predispose the macrophage to take on that phenotype, or it's completely driven by the muscle cell that had the mutation?
So we have a paper in submission on macrophages, like from iPS derived, same cell lines, just with made macrophages versus control. There are differences in their endocytosis, not phagocytosis. There are no difference in polarization. There are a lot of transcriptomic differences. Some of them relate to metabolism, and definitely there is a difference in their co-interaction with muscle interstitial cells. So, and secretome. So definitely, you know, they have different secretome, at least, you know, in vitro, and so likely contributed to the disease.
Sorry, like the one last very just clarifying question for this. Again, go back to story 2. The nicotine activates sympathetic nerve. Then you see that the increasing the beating of the muscle. Is that just the nicotine activated sympathetic nerve? Then the sympathetic nerve through the neuromuscular junctions activating the muscle cells, or there's also secreting something that increasing?
Well, when you activate with nicotine, the sympathetic neurons, they basically secrete norepinephrine. And that's what activates the cardiomyocytes, usually, the beta-adrenergic receptors. And so and some alpha, but not much in cardiomyocytes, more in the glial cells, which we don't have. Which we have in 3 culture, but not in 2 culture. So, yes, if you specifically block norepinephrine with some drugs to be released from sympathetic neurons and add nicotine, you don't see the effect. So this tells us the specificity that goes to norepinephrine secretion. Good questions. Thank you very much.